Welcome to the virtual roadshow series presented by Channelchek and Noble Capital Markets. Of the 6,000 small and micro-cap companies listed on Channelchek, today we're featuring Lineage Cell Therapeutics, NYSE American ticker symbol LCTX. Research coverage of Lineage Cell Therapeutics is provided by Noble Capital Markets. Noble is a FINRA-licensed, SEC-registered broker-dealer. All Noble research is available on Channelchek. Following the formal presentation, Ahu Demir, Noble's biotechnology research analyst covering Lineage, will ask our presenters a selection of the questions received during the broadcast. And now, here is Brian Culley, CEO, and Brandi Roberts, CFO, of Lineage Cell Therapeutics to make the presentation. Brian and Brandi, take it away.
Well, good morning, good afternoon, and thank you, everyone. I look forward to being able to present the Lineage story to you today. As Lineage is a public company, Brandi and or I may be making some forward-looking statements, so we'll refer you first to our safe harbor clause and additional information about our risk factors at the SEC website. Brandi and I have extensive experience managing public companies, biotech companies. We have run some very large clinical trials in the past and are just delighted to be relatively new management to Lineage. I recently crossed my 2-year anniversary, and Brandi joined shortly after me, and we think we've done some really wonderful things with the company over the last couple of years. So just to provide you with a quick overview of the organization, Lineage has a very innovative approach. We are a cell therapy company. However, what we do is we transplant differentiated cells. So we do not put stem cells into any patients. Stem cells to our company are just starting material. Stem cells are special because they have the ability to duplicate or replicate without changing. And so we can take those cells, we can divide them and increase them in number, and then we can direct their differentiation, meaning we can manufacture specific and unique cell types which exist in your body. Then we use those specific cell types as therapeutics to treat various conditions and diseases. And we'll go through examples of what we do in just a moment. We have world-class expertise in intellectual property. We have many hundreds of patents applications and issued patents around the world. And we also control our own manufacturing facility. And we today have 3 clinical stage programs. Oprogen is our phase 1/2A clinical study for dry age-related macular degeneration. We manufacture retina cells to treat dry AMD. We also have a program called VAC2, which is an oncology program. So we manufacture a special kind of immune cell to help fight tumors. And then thirdly, we also have OPC1, which is our spinal cord program. And in this case, we manufacture a different kind of cell. We manufacture what's called an oligodendrocyte, and that is used to treat spinal cord injuries. The beautiful thing about cell therapy is that it has attributes and it has the potential to do things that traditional approaches cannot. And we've begun to see evidence of some of these special and unique attributes. So for example, we recently reported the first ever case of retinal tissue restoration in a patient. So we were able to salvage or recover retina tissue. And that's special and unique because that's something that humans are not able to do. They can't grow retinal tissue, but we were able to grow retina cells and transplant them to the body and actually restore some of the dying tissue. We've also been able to add cells into an area of injury in a spinal cord, which ordinarily would form a cavity or a hole. And we've been able to put our cells there and they divide and they occupy that space. And we've seen some very exciting recovery, mobility in patients who are otherwise paralyzed. And I'll talk about some of that today. And we also are able to manufacture a third kind of cell, a dendritic cell. Dendritic cells are important for vaccination, whether that's vaccines against tumors or vaccines against infectious diseases. These are all multi-billion-dollar or each individually billion-dollar-plus opportunities from a commercial perspective. And we have a lot of important catalysts. In fact, in just about a week or so, we have a significant update on our macular degeneration program. And a few weeks after that, we're going to provide a manufacturing update on our spinal cord program that a lot of people have been waiting for. So over the next just month, we've got a couple of pretty significant events. And the company has a strong cash position. We had about $38 million as of September of this year. So our technology, as I shared, is a little different. We're trying to usher in a new branch of medicine based upon the manufacture of specific cell types and transplanting those cells to the body. This is what that looks like. These almost poached egg-looking things with the blue center. Those are pluripotent cells, meaning those cells can become any of the cells in your body. They can become bone or hair or kidney or liver or heart. There's a recipe that they follow, or an instruction plan, which those cells follow in order to become specific cell types. And we know those recipes in some cases. So we have the ability to take those undifferentiated stem cells, those blank slates, and we can direct them to become pure populations of retina cells or spinal cord cells or dendritic cells, and then use those specific cell types to treat the body. We don't genetically manipulate the cells. We just treat them with different chemicals and compounds in order to give them the instructions as to what kind of cell to become, but we do not manipulate their underlying DNA. And that's really important from a safety perspective. The other nice thing is that we have already gotten to pre-commercial scale. We have the ability to manufacture incredible numbers of these cells, and I'll share some of that with you in just a few moments. This is an image of our manufacturing facility. We control all of our own banking and handling. We do all of our scale up. We've got multiple clean rooms to be able to generate our clinical trial material. And as I said, we've got hundreds of cell therapy related patents to protect our products if and when they reach approval and commercialization. This is an image of our current clinical stage pipeline. So these are just the clinical stage programs. What's notable about them is that each of these programs has received significant external funding in the form of grant support or in-kind support, whether that's from the Israeli Innovation Authority or the California Institute for Regenerative Medicine or Cancer Research UK, one of the largest charities in the world. There in the green squares, you can see significant investment in these programs from very credible groups. And these programs are all reaching important clinical milestones. We expect to finish enrollment in the AMD program this year. We have completed a study in the spinal cord program and are working on manufacturing, and I referred to an update that's coming in the next few weeks. And we just have 2 patients left to enroll in the Oncology program. And I should mention that $14 million that we received from the California Institute for Regenerative Medicine. There was a ballot initiative in California, which appears to me to be likely to pass. It is going to be narrow, but it will refund CIRM with more than $5 billion of new capital to support stem cell research and stem cell product development and facilities. And it's very exciting for the whole field. It's certainly exciting for Lineage as a stem cell company, but it's also exciting for other companies. And we're really happy to see that ballot measure looks like it is on the way to being approved by the voters in California. So first, I'm going to talk about OpRegen, which is our therapeutic approach to treat dry AMD. Dry AMD is a horrible condition. It's one of the leading causes of blindness in the world. And the feature is that there are areas in the back of your eye where specialized retina cells reside, and they can begin to die off. And you can imagine this is like an aerial photo of a forest fire, just expands outward, more or less in a circular direction. And that's what these 6 panels are showing from 2012 to 2019. This area of atrophy, the area of dead cells, is just getting larger and larger. And if you follow over time that red line, that's a readout of the patient's vision. So you can see that they've gone from having normal vision with a very small area of damage, that as that area of damage got larger, this patient became legally blind and then really had no vision to speak of whatsoever. And that is because those retina cells have died off. This is a massive commercial opportunity because most people who suffer from macular degeneration suffer from the form I just described. They suffer from dry AMD. There's another form called wet AMD, and it's doing $10 billion in sales already, but the dry form has 0 FDA-approved products. So this is completely untapped commercial opportunity for companies like ours and others. How we aim to capture this opportunity is by the manufacture and the transplant of retinal pigmented epithelium cells. So on the left cartoon here, you see these cells that are shaded down, they're darkened. Those are dying retina cells. That reflects the images that I showed you. And when those cells die off, there is waste material shown here in yellow, which accumulates. And also the photoreceptors, another specialized cell type, they begin to die off. So our view is that if you can manufacture and transplant brand new retina cells, you can clear out that waste material that accumulates. You can salvage and rescue those photoreceptors, and you may be able to provide improved vision, or at least be able to halt the progressiveness of this disease. We've seen some very encouraging signs in our phase 1/2A clinical trial. One of the big fears that people have about cell therapy is that the cells are going to get into the body and either they're going to be rejected or they're going to begin to divide and cause tumors. We have seen none of that. We have had 0 cases of acute or delayed rejection of our transplanted OpRegen cells, and we certainly have not had any people growing tumors in association with our treatment. What we have seen are very encouraging signs of an effect stemming from our transplant of the cells. So we are seeing patients that are able to see better, their areas of atrophy are growing more slowly, we've seen a reduction in the waste material, and we've seen durable engraftment. These cells are resident for multiple years, so it's possible that this may be a one-time treatment for these patients. Most important of everything, we also recently announced that we had seen that restoration of retinal tissue. No one has ever seen that before. We're the only company to report it, and we're really excited to continue investigating it and see where that path leads. Looking at visual acuity, I'm going to focus just on cohort 4 patients. That's because the first 3 cohorts which we tested, those were patients that were legally blind. So they had very advanced disease, and we didn't really expect that we were going to be able to do much in terms of vision. And in fact, there's some market research that's been done by another company showing that most retinal surgeons expect that the benefit that may be attributable to these treatments would come from patients that have better baseline vision. So if we focus on patients that have better baseline vision in our study, we're seeing a separation of these curves. We're seeing that the treated eye is able to read more letters after treatment compared to baseline than the contralateral eye or the untreated eye. It's still early. There's only a relatively small number of patients here, but we're really excited that we're off to a good start. This is an example of what a 10 to 15 letter improvement in vision could look like to a person. So if you can move 10 or 15 letters, you might be getting someone's driver's license back. You might be enabling someone to be able to really use a cell phone without a lot of difficulty. And those are important quality of life metrics. People really care about their vision in a way that the other senses don't really enjoy. So we really are working to try to bring this kind of a benefit to patients. Now, with respect to the disease condition itself and the progression of that area of atrophy, this is an overlay of an area of atrophy, a before and after, 9 months apart. The red shaded area, which is covered now by the blue shaded area, but you can see that the red shaded area is larger. That is the baseline area of atrophy. So that is where the retina has begun to so it's sort of like dehydrating or collapsing. And after treatment with our cells, we were able to show that that area of atrophy was actually smaller. And you can see it through all sorts of different cross sections. It's not just the aerial view. You can look at dozens and dozens of cross sections and see that the outer nuclear layer and the other important layers, I won't get deep into the technical side, but what's important here is that we sent these data out to several experts in retinal imaging, and they were convinced that what we had shown was authentic, and they encouraged us to try to repeat it, which is what we're trying to do. We have seen that the rate of atrophy growth has been slower in some of our treated patients. It's not a massive difference, but again, it's a small and directionally encouraging piece of data. And we're going to do a significant update. This is just on a handful of patients. This is from a data update, which we did a number of months ago, 4 or 5 months ago. The next update is just about a week away. So you'll be able to see more patients contributing to this data set very soon. We also are working to deliver the cells in a better way. So we don't just rely on our cells to be able to provide this benefit. We're trying to find better ways of placing the cells. And so the traditional method is you push a needle through the front of the eye and you puncture the retina and you discharge the cells. The problem with that method is that when you remove the needle from underneath that hole that you formed in the retina, the cells can leak out of the area and go into the wrong areas. And that's not something that we want. So we have an exclusive option with another company which owns the right to a subretinal delivery system, which allows us to go around the outside and the back of the eye, bring a needle up beneath the retina without puncturing it, place the cells under the retina or in the subretinal space, and then retract the needle. So overall, when we look at the competitive landscape of dry AMD, there are a number of different approaches. There's cell therapy, complement inhibition, and other things. But only Lineage is the company that's been able to show retinal restoration. Only Lineage is using the Orbit SDS device to deliver cells. And only cell therapy has the promise of being able, or the potential to being able to deliver through a single treatment, or maybe every few years, a single administration of cells that have a long and durable effect. So we believe we're well positioned among the competitive landscape. And in fact, we could probably coexist with a lot of these approaches. These are companies that may be able to slow the progression of the disease, but they are not likely to be able to stop the disease. So they're not going to be able to get patients taken away from us, so to speak. Continuing on, if we are successful, we're going to need to manufacture a lot of cells. And we have worked very hard to shift from two-dimensional cell growth to three-dimensional cell growth. And we can, in fact, now manufacture 5 billion retina cells in a 3-liter bioreactor. So that's about the size of a milk jug. And that equates to about 2,500 clinical courses in one of those containers. And you can do a bunch of those containers simultaneously, or you can put them in series or parallel. And this is a true thaw and inject formulation. So this is an allogeneic therapy. This is an off-the-shelf therapy. We are not starting with a patient's retina cells, taking them out, growing them, and putting them back in, or taking them out, manipulating the DNA and putting them back in. That's not what we do. We have a pluripotent cell line that allows us to manufacture billions upon billions of cells. They are frozen down. When they get to a clinical site, they are thawed in just a few minutes, put into an injection chamber, and delivered to the eye. This is an update on our clinical program. As I said, the first 3 cohorts, those patients were all legally blind. We are now currently in cohort 4, and you can expect a significant update on our enrollment soon, certainly no later than the AAO annual medical conference. It's a major medical meeting coming up November 14th, 15th, 16th, right in around there. And we also are going to have a KOL day where we have one of our investigators talking about our program in a 1-hour webinar, not unlike the one that we're doing today. So overall, I think we're well positioned. I probably have mentioned all of these points, other than we may be able to go into additional disease areas, and we have an opportunity, of course, to consider strategic partnerships for this program. But overall, there are a lot of positive things that directionally to us, we feel very good about continuing on, trying to demonstrate that OpRegen cells, a suspension of RPE cells, can provide a benefit in patients who suffer from age-related macular degeneration. I'm going to change now to a different topic, but I'm going to go a little bit faster through these next 2 diseases, not because they're not important, but because you basically understand now our approach. So we're doing something similar with the spinal cord. We're doing this for people like Lucas Linder. He was driving that truck that you're seeing. A deer jumped in front of his truck. He was not wearing a seatbelt. He was ejected from his vehicle, and he was paralyzed. Could not move his arms and legs, could not move his shoulders. He was a participant in our clinical trial, and as you can obviously see, he has regained significant upper extremity mobility. We have people who are able to type 30, 35 words a minute who previously could not move their fingers. So you can see what an amazing clinical benefit and opportunity we may have if we can provide these individuals with some clinical benefit. The approach here is that we manufacture a different kind of cell. Instead of retina cells, we manufacture oligodendrocytes, and that's a special kind of spinal cord cell. We inject those into the spinal cord. This is an orphan disease, so we have orphan drug designation. We have RMAT designation for this program. And as I said, we've received significant financial support from the California Institution for Regenerative Medicine. And what we're trying to do here is take an injury that has occurred to the spinal nerves here in your cervical neck area, and those are the nerves that are responsible for your upper extremity mobility, your fingers, your wrists, your arms, and we're trying to help that area heal, and to do so in a way that it provides the connections between your brain and your finger, and those are the connections which direct mobility. So it's not enough to just grow axons. Those axons have to be myelinated. They have to have the insulation, which allows connections to be connected. And that's what our oligodendrocyte cells do. They provide that remyelinating activity. They provide the myelin basic protein, which is required as insulation, because nerves that are present but are not myelinated, they won't function properly. What they say in the field is that nerves that wire together fire together. So in order to get reinforcement so that you can have a clear message, I want to clasp my fingers tight into a fist like that, that requires not only the cells and the axons, but all the supportive information like the myelination, also the physical therapy. That's an important component as well, and that's part of what happens with these patients as well. We are just on top of, we are additive to all the things that these patients would be doing. You can see here, I'm just going to focus on the three approaches, three ways that were beneficial. We can prevent the cavity formation. So we prevent that whole cavity, just like in your tooth, cavity in your spinal cord. It's a really bad thing. And I'll tell you, electrical impulses cannot jump across gaps. So this is a very important feature of our technology, that we fill in that gap. I jokingly say that it's like spackle for your spinal cord. When that spackle is in place, it helps to remyelinate those axons and secrete neurotrophic factors. Altogether, we think this is what's driving clinical effect, or what should be able to drive a clinical effect in patients. There are 25 patients who have been treated with this therapy. On the left, those first 3 patients, they got a very subclinical dose. So we did not expect anything meaningful with only 2 million cells. So we focus on the 22 patients that received either 10 million or 20 million cells. That's the clinical dose for us. And importantly, this is not like stroke treatment. You don't have to be at the diving board or in the surf where the patient has suffered the injury. In fact, putting our cells in that quickly, it's a hostile environment. There's a lot of inflammation back there. So we wait until between 3 and 6 weeks after the injury. That's the right time for our cells to go into the patient. And that's also enough time to make sure that the patient is stable, they're a good candidate for a clinical trial, they can provide informed consent, and so on. What's been very exciting is that almost every patient, 96% of them, had a successful engraftment, meaning the cells were placed and they remained durable for years. So they were not rejected, just like in the eye, they were not rejected. We also saw approximately a third of patients, 1/3 of patients on this study had 2 or more gains in their motor level. That is a very significant increase. I'll show you what that looks like. Gaining 2 levels is like moving from C4, right, to C6. You're pushing the injury down. And when you push the injury lower, you're able to pick up mobility in the upper extremities. So going from C4 to C6, and we had patients who did this, is like going from around the clock support to being able to bathe yourself, dress yourself, feed yourself. You can see the massive change in quality of life by being able to just get a little bit of functional improvement in these patients. The clinical data continues to demonstrate that this is really looking to be a safe and well-tolerated treatment. We haven't had any complications associated with the OPC1 cells, and patients have had durable benefits. It's been wonderful to see that no one has had a decline in their motor function. Certainly some people do have spontaneous regeneration, and so what we are planning on doing next is a comparative study to show that patients treated with OPC1 have better outcomes than patients that aren't treated with OPC1, and we believe that that could be the basis for a registrational study or a study for the basis of approval due to the unmet medical need and the orphan drug status of this and the no alternative therapies for this patient population. So importantly, the early clinical data was not supported by a lot of, the early clinical data was not supported by the same level of excellence on the manufacturing side. So for the past year, a little more than a year actually, we have been working quietly behind the scenes on the manufacturing side of this program, because the early manufacturing, which led to the clinical studies, that was just enabling, just to make sure you could get into a clinical trial. Now that we've seen encouraging clinical data, it was the right time to work on the manufacturing so that you can actually have a product. If you want to run a clinical trial of any kind of large size, you've got to be able to manufacture a lot of cells. And so we took advantage of our experience in cell manufacturing, and we took advantage of the success that we've had with the retina cells, and we asked our team, our internal team, to work on the OPC1 cell manufacturing. And as I mentioned, we're going to have a major update on that in just a few weeks. So the clinical trial that we've had, we've been really happy to see that the mechanistic proof of principle is intact, meaning that after we treat the patients, we're able to interrogate their immune system and see that they have T cells which are reactive to our antigen. So we put the antigen in and we ask it to inform the immune system, and then we ask the immune system, did you get the message? And we have evidence that it is getting the message, and sometimes it's getting the message loudly and clearly. So, upwards of 2-3% reactive T cells in patients, which is an extremely high number. So this is a very nice validation of the mechanistic approach, and we intend to collect additional data on this and really start thinking about where we might go longer term with this as a platform. Just briefly, we had mentioned about the financial situation. The company's market cap, it vacillates. It's a little below 200 million. We have about 53 employees. We try to be very economical with our spending, and I think that reflects a commitment by the new management team to deploy capital in the smartest and best way possible. Again, I really want to orient, clearly there's a lot of events coming up for us. I mostly want to orient you to the AAO Medical Conference and our interim update on our dry AMD program. That's ahead November 13th to 15th. Also, we will have that KOL call on November 17th. And then in early December, we'll do the OPC1 update. So very important things for people to keep track of. So Lineage is an early and emerging leader in allogeneic cell therapy space. We've got enormous intellectual property portfolio. We have very commercially valuable product opportunities, and I think we've been making excellent progress with our clinical programs to date, and we intend to continue doing so. So last slide here. We obviously are motivated by the patients. This is who we serve. This is who we're trying to develop these programs for. And we really welcome you to take a look at the content that we make available on the media page of our website and learn more about the kinds of experiences that our patients have had on our clinical trials and with their diseases.
Thank you, Brian and Brandi, for joining us today. I'm Ahu Demir, the Noble Research Analyst who covers Lineage Cell Therapeutics. And here are a few questions I have selected, as well as a couple of my own. So Brian or Brandi, my first question would be, what would you suggest as the most important endpoint for treating dry AMD, and what should we be looking for to see for a registrational phase 3 study?
Yeah, there are a couple of questions. There are a couple of ways to answer that. So the FDA seems comfortable with using growth rate of GA, so that area of atrophy in the back of the eye. Companies have tried this in the past, and several are continuing to do so. The reason why they don't choose vision is vision's tough. It's really hard to show an impact on vision because this is a very serious disease. By the time you've got cells dying, there are a lot of things going wrong back there. So a lot of companies are happy that the FDA is not insisting that you have to show an improvement in vision, but is willing to accept that if you can slow that growth rate of GA, that's going to be a meaningful clinical benefit to the patient. Because clearly, when that GA gets big and covers the macula, you're losing your vision. So those are two places that you can go. You can go through best corrected visual acuity, or basically how many letters can you read on an eye chart, or you can look at growth rate. The other thing, which is new, and I think it's only relevant to us, and we haven't talked to FDA about this, is what if you did a responders analysis based on patients who actually had a reduction in their size of GA? Now, I'm being a little prophetic or a little ambitious, but we have shown it, and I'm hopeful that we'll be able to show it again. So, in theory, there's a third endpoint that one might consider in this space, which is showing some percentage of patients who actually have a smaller GA over a period of time, not just a slower growing GA.
Thanks for your answer, Brian. So my next question will be again on the AMD program. What do you think has been the main obstacles in getting approval for these patients, considering there are no approved products currently in the market?
Yeah, why with billions of dollars available, why hasn't anyone gotten anything approved? Not for lack of trying, certainly. I think the problem is that we don't know what is happening in this disease. We don't know why these cells die off. Clearly they do, and there are a bunch of molecules that are associated with that process, but trying to find a single causal effect is very difficult. And most traditional approaches in medicine today involve a targeted approach, hit one pathway. It's very narrow, and that can be beneficial because if you're narrow on one pathway, you might avoid side effects. But if you've got an avalanche coming at you and you're trying to just like pick out a couple of snowflakes, you're not going to have a difference. So our view is that the only reason that we haven't been successful in this disease is that we're trying to hit just very specific pathways, and that you might get better results if you become indifferent and you say, look, I'm just going to give you a whole new cell. I don't know if it's pathway A or B or C or all of them all together. How about you just have a new cell? And so I don't know, it remains to be seen, but I think that that's probably one good reason why we haven't been successful in dry AMD as an industry yet, is that we're too narrow with our approaches.
And as we talked about the high-level approach on the indication, if we would get into AAO data next week, what are the expectations? What do we expect? Are we expected to see additional data from more patients, and how would it impact the program going forward?
Yeah, it's been a number of months since we've done an update. So we'll have an update on enrollment in the OpRegen study. We'll have an update on safety with more patients that we had in the past and longer follow-up periods. We'll have an update on efficacy, again, with more patients than in the past and with longer follow-up. And it's a slow disease, so longer follow-up is meaningful to us. And we'll also have an update on the use of the new delivery device. So those four items will be important for everyone to look for in a week and a half.
If we get into the next program, OPC1, the first OPC1 trial was funded by California Institute for Regenerative Medicine, and you mentioned that Proposition 14 in California is likely to pass. Could you elaborate on what it would mean for OPC1 program? If this initiative is approved, would you intend to return it to CIRM to further develop OPC1 program?
Yeah, let me just invite Brandi to answer that, just so we, get both of us involved in the Q&A today.
Sure, thanks so much. Yeah, we absolutely would intend to go back to CIRM and ask for additional support. I think if you look back at the records, OPC1 was one of the first programs that CIRM ever funded, and if you do a search for Prop 14, you'll see a couple of our patients from our study on that website, because everyone was very happy with how they performed. And so we're obviously watching with great attention to see if it will get approved. I think we're at about 86 or 88% with the ballots counted right now, but it is passing at this point in time, and we'll keep an eye on that. And fingers crossed, if it does get passed, we'll definitely go back to them for the future needs.
Sounds great. And still going on to the OPC1 program, we received one question regarding what other indications you could go after using OPC1 program. Is MS indication a target? And are you searching for partners or CRM funding for MS application or beyond?
Yeah, that's a very smart question. Thank you for whoever sent that in, because we sort of get talked about like a spinal cord program, but it's not. What we have is an ability to address conditions of demyelination. So that's how we view a spinal cord injury, is that you've got a wound there, and you need to remyelinate in order to reestablish the connections. And so there are other conditions for which the main feature or hallmark is demyelination. So multiple sclerosis, white matter stroke, there are others. So if you think again of a life cycle management or a long-term strategy, getting to market on spinal cord injury would be fantastic. But having an ability to expand into other areas when we already would have safety data, we already would have commercial manufacturing, that's just very wise of us. And we actually have done some preclinical work in other areas like MS and stroke, exactly to explore the use of these cells in those settings. So we're not close to the clinic in those areas, but they are things that we're aware of and think about longer term. With respect to partnering, it's been our strategy to, since we've joined, to alert potential partners about the work we're doing. I think it was not urgent for us to get OPC1 in front of partners for the past year, because we knew that there was this manufacturing deficiency. A partner would have great difficulty going into the next clinical trial with the manufacturing in the way, shape, and form that we received it in, or that we purchased it in. But we've done work in areas like scale and purity and quality and testing and so forth. And so I think now we're kind of approaching. I don't want to steal thunder from an announcement in a few weeks, but we're approaching a point at which being able to take the combination of the clinical data that has been generated to date plus manufacturing achievements and say, hey, this is a nice package. Ultimately, we may or may not elect a partner, right? That's a real shareholder. You got to wait till you have your terms, and you wake up one day and something's been partnered. But we have been talking, and we will continue to talk to potential partners about each of our programs, frankly, because it's always prudent to do so. It gives optionality.
That's certainly true. So switching gears to vaccine program, Brian, it appears that it can be used as an adjuvant. And yesterday we got this data from BioNTech and Pfizer. Would you think you can use vaccine in combination with any of these RNA vaccine treatments for coronavirus infections?
Yes. So the BioNTech data is interesting to us because there you're talking about using RNA and administering it to a patient, and you're sort of hoping or expecting that material, in this case the spike protein, will be picked up by dendritic cells and presented to the immune system. So what we can do is we can skip that. There's less risk by putting that mRNA already into the dendritic cell, right? You're skipping that pickup step, that pickup and expression. We could do the pickup and we could do the expression, and that, to our minds, gives us a greater level of confidence that the message, right, the foreign message, the antigen, would get noticed by the patient's immune system. And I can tell you, as I was saying, it's funny because I was looking at these slides just recently. Let me look at this. So when BioNTech and Pfizer, they had some ELISpot data looking at the T cell response, and it was 0.02 to 0.04%. So that's a percent of T cells that are responsive to the antigen. We were like 50 times higher than that, right? Same assay, ELISpot data. Now it's cancer, it's a different antigen, and so you can't make an apples to apples comparison, but it does not surprise me that 0.02, 0.06, which is the Oxford vaccine, 0.04, which is the high end of the vaccine for Pfizer, those are like successful levels for a naked RNA or an adjuvanted RNA approach. If you prepackage and you put that RNA into the dendritic cell, you can get 10, 20, 50 times higher levels of reactivity. Now, we don't know the clinical relevance of that, but it seems like it would be a good thing. So, the short answer to the question is, yeah, I think we could do that. Our focus today is on oncology, but funding time and et cetera, I think mechanistically, biologically, it's a feasible approach.
That makes sense. And Brandi, I will just refer my next question to you. So could you talk a little bit about the balance sheet, quarterly burn, and also maybe some information on how well capitalized you are to achieve meaningful milestones over the 12 to 18 months, considering you have multiple catalysts coming up?
Sure, happy to do so. So we actually just had our earnings call last week, so we talked a little bit about this then. We reported cash and marketable securities of about $38 million as of the end of September. Included in that balance was a payment of $24.6 million from Juvenescence for the sale of some Ajax shares back in 2018. And we did get that payment as expected. So, we've really been able to capitalize this company, very well using our marketable securities. We've been able to make smart decisions about when to sell those. We haven't had to do a traditional equity financing in over 3 years. While we've worked on, essentially transitioning this company into a preeminent cell therapy company and having 3 programs in clinical trials. Remember that we did the Asterias acquisition back in 2019. If you look at those companies separately in 2018, the spend was about $43 million at that time if you combine the companies together. We're now at less than half of that. Our burn for the first 3 months of 2020 was less than $15 million. So we have made tremendous progress in bringing down our operating expenses. We continue to evaluate what makes sense. We actually just terminated our leases up in Alameda and took a smaller space that will enable us to save $780,000 over the next couple of years. So we have a pattern of continuously looking at our spend and what makes sense while we're pushing these clinical cell therapy programs forward. We feel that our funding right now is good for well into 2022. We're focused on getting to the next milestones, many of which Brian already talked about. We're looking forward to the AAO update. We're looking forward to the completion of OpRegen enrollment. We're looking forward to the VAC final 2 patients getting dosed as well. And then we also have an OPC1 manufacturing update coming up in just a few weeks. So lots to look forward to.
Thanks, Brandi. That brings me to my last question. Actually, you answered partially, but what makes you excited about working at Lineage, and what should investors be paying attention to the company right now would be my last question.
Wow.
Sure. Go ahead, Brian. You first.
I think cell therapy, which has been promising, amazing outcomes for a very long time. I think it's finally here, right? The era of cell therapy is finally here. We've seen it kind of happening on the oncology side with T cells and everything, but now we're finally seeing it happening on the allogeneic side, which is where you get the right economics for manufacturing. You're seeing it because it's being viewed as a transplant approach where it can accomplish things that traditional approaches cannot. And programs are moving through the clinic. It's not just us. I mean, there are other companies that are working on other organs and other systems that are having a lot of success. And so I think this is an incredibly exciting time. We appear to have a new administration that I think is going to be very favorable and supportive of a scientific view in the world. That's going to be great for biotech, and I think in particular for emerging technologies like cell therapy.
Yep, and I'll just add to that. I mean, obviously we have amazing commercial opportunities with all 3 of our programs. And I would just add the ability to potentially impact so many patients. As Brian mentioned, we have lots of stories about patients with dry AMD, with spinal cord injuries. And if we were able to make a difference for these patients, I think it would be just truly amazing to be part of. So really, really excited to be here.
Sounds great. Well, Brandi, Brian, that's all the time we have today. Thank you for helping us get a better understanding of the company's story. If you would like to get the independent research I have written on Lineage Cell Therapeutics, go to channelchek.com and type the company name or the ticker LCTX in the search box located in the top left companies section of the site. And again, thanks to our guests, Brian Culley and Brandi Roberts from Lineage Cell Therapeutics.
Thanks, Bye, everyone.
Bye-bye.
This has been the Virtual Roadshow series brought to you by Channelchek and Noble Capital Markets. Log into Channelchek or subscribe to our YouTube channel to get the full catalog of roadshows and C-suite interview channelcasts. Register for Channelchek's no-cost premium content and receive notifications of new releases from both series. Thank you for joining us.